So the way this is going to work is our topic for today is single cell analysis, and I'm going to do a little bit of an introduction, tell you why why we do single cell genomics. I guess at around 11, or probably maybe we'll push it back a couple of minutes, we're going to have a little discussion. I'm going to lecture now this morning. Tara and I will lead the discussion. And then after lunch, Tara's going to do a methods lecture focusing on really sort of two fundamental and important tasks that take place when you do single cell analysis, namely normalization and linear regression. And she'll walk you through like some of the methods and the math behind them. And then you'll do a thing on Google Colab where you actually practice. Okay, so that's kind of the outline. So let me go back in here and right. So to motivate like the neuroscience interest in this, I'm just going to use. There's many. I could say a whole. Yeah, you need to listen. Sure.
Maybe without.
You can clip it on my shirt if you want. You want me to take this off?
Yeah.
It's nice. Mm-hmm. Great, thank you very much. So to motivate this, I'm going to talk about this project I'm involved in. One could talk for much more than an hour about motivating single cell genomics. But this is a project that I got involved in several years ago. Actually, thanks to David Anderson, who has a long-standing collaboration with the Allen Institute, who themselves got involved in this. And this is so the Brain Initiative is a large project run, I guess, seeded by President Obama. And within that project, there's a sub-project to study the mouse and then maybe some non-human primate brains and a little bit of human brain using single-cell genomics technologies. Within that project, and so within that scope, there's something called the BICCN, or the Brain Initiative Cell Census Network. And within that, the primary assay being employed is single-cell RNA-seq, which I'll tell you about today. And the goal is sort of to map out the different cell types to first order in the mouse brain. So I'm not going to go through this at all this morning, but just so you know, there was an entire, this is a cover of an issue of Nature, and the entire issue, it's from, I think, October. So it just came out, actually, like a couple of months ago, 6 months ago, I guess 7 by now, right? And the entire issue is devoted to the brain, and there's lots of pictures like this of atlases. This first analysis that constitutes this whole issue of Nature is on the primary motor cortex, so only. Currently, this project is scaling to the whole mouse brain, and there will be another probably issue of Nature next year in which that will be published. And that's quite exciting, actually, for you, maybe, because all of this data is publicly available. And I can tell you that the consortium, which is a huge number of people, just scratching, not even scratching the surface of these data. They're like peppering a few sprinkles into Nature, but there's like so much to study here and learn here. And whatever you're working on, there's probably data in here that's interesting. Okay, so I guess our goal, Tara, and my goal is to like have you today learn a bit about how, like what the data look like that generate these sort of pictures, how these are made. I can't promise that by the end of the day you can do all of this yourself, but maybe you will at least have an appreciation for the steps that are involved. And if you don't, then we're going to stick you in this closet over here. This is a secret room back here. No, I'm just kidding. All right. I'm just going to close it because I'm sort of nervous something will walk out of there. I've been watching this show, Stranger Things. Okay. All right. Okay, so I don't have the slides in front of me, so I'll just, that's why I'm looking over here. But what you get out of one of these experiments is, and you already probably all know this, is you get a big gene expression matrix. We'll talk a little bit about what that is. The cells can be viewed as points in high dimensional space, and you already heard about that last week. And what people do with this often is they cluster these points to define putative cell types. You make plots like this, and you try to understand, what both what are the cell types maybe that you're looking at, and also what genes mark these cell types. And all of these sentences carry a lot of interesting depth to them in terms of what it really means to do these practices. And that's what I'll talk about. So first thing I want to do is actually motivate why one might do single-cell RNA-seq. I think it's useful to go through this because, it's true that with every new technology, it's kind of fun to deploy it, but it's also true that when you are actually performing an experiment, you should use the technique that's appropriate, not the technique that exists. And so single-cell RNA-seq has a history. It's derived from bulk RNA-seq, which itself had a precursor in microarray technology analysis, and that was there probably before you were born, like 20 years ago. Was a popular technology. There's several things I want to say about that, is that the microarray data is not so great. It's very noisy. There's a lot of it available. But the methods that were developed in analysis of that data, many of them really underlie the methods used today to do single-cell RNA-seq analysis, or just more generally single-cell genomics analysis. So it would be wise, if you're interested in these technologies, to know about this literature. And I can give you some pointers later today. Bulk RNA-seq, gives you just one vector, that is like one measurement for each gene in a pool of cells. And there's a lot of bulk RNA-seq data out there in public databases. This is a plot from a paper just showing you like how it's taken off over the last couple of years. It's still widely employed. And I might remark to you that it was developed in several places simultaneously, but the primary place where this happened was actually at Caltech. So by Barbara Wald and her trainee at the time, Ali Mortazavi. So this is sort of just good to know. All right. So why do people do single-cell RNA-seq? And I just want to go through 2 or 3 slides about this in case you're not familiar with it. And that's this idea that averaging can be really problematic. And there's a sort of, there's several words that are used to describe this issue. They're called Robinson's paradox or Robinson's reversal or Simpson's paradox. I'm just going to tell you what these are. So this is cartoon. It's just a sort of thought experiment for you to think about. If you were looking at the expression of gene A versus B, you might see a sort of relationship like this, even in a single-cell experiment, where you have a sort of negative correlation. That is, the more you have of gene B, the less it appears you have of gene A, or vice versa. But in fact, if you were to actually know how to group the cells, you might find actually positive correlation within each group. And this is called Robinson's reversal when that happens. It's actually interesting that this actually happens in real data. One of the things Charles looked at is that exact issue. And but anyway, this kind of motivation comes from a nice review. It's from my former student Cole Trapnell. It's part of the motivation why it's important not just to do single-cell genomics, but also to identify the classes, because you may be misled as to the relationships. There's another, so this reversal is a sort of continuous phenomenon. Like the gene A and B expression here, however they're measured, there's some real numbers, there's some numbers. But there's a discrete analog of this that also is important. And if you haven't seen it or thought about it, it's really worth doing. This actually came up recently in COVID. I should update this example. I like this one. It's from the, these are the results of the vote for the Civil Rights Act in the 1960s. But this just comes up all the time in daily life, so it's good to think about. So the issue here is like this. So this is an example from the Civil Rights Act. So you had a vote by Democrats and Republicans in both the House and the Senate. House is up here, Senate is below there. And well, the Democrats and Republicans at the time didn't map exactly onto Democrats and Republicans today, okay? But regardless of that, you can see that among the Democrats overall in the House, 61% voted for the Civil Rights Act. Among Republicans, it was 80%. But actually, if you break it down by Northern and Southern Democrats, you get the opposite. You find that 94% of Democrats in the North voted for it versus only 85% of Republicans, and 7% of Democrats in the South versus 0 out of the 10 Republicans in the House. Same happened in the Senate, okay? So this is it's sometimes called Simpson's paradox because it appears paradoxical that when you divide into groups, North and South, you have one effect, but when you average together, it's reversed in proportion. But you don't have to, it's not a paradox, it just can happen, and you don't have to take my word for it. You can just actually calculate the numbers here. So these are the actual votes. This is really important in life in general because, for example, in politics, the parties use this all the time. Every election cycle, Republicans on Fox News will claim that they voted for the Civil Rights Act and the Democrats didn't, for example. It's the same with your single cell data. If you have not actually correctly curated the groups of cells, you can have a completely different impression of what actually is going on. And sometimes it's important to average and sometimes it isn't. If you'd like to just understand what's going on here geometrically, basically, so I have V1 and V2 and W1 and W2. So I have two groups here, group 1 and group 2. And you can see that, there's the yay votes and the nay votes, if you want, on the up, one axis this way, yes on this axis, no on that axis. And what I'm computing here, these percentage, it's like I'm taking yes over the total, right? So actually, if you want to think about it, this is maybe yes, and this is the total. That doesn't matter, but let's say yes and total. So what it means to compute a percentage like 94 is means I take this value divided by this value, which is the slope of the line, okay? So what you can see is that I can have that V1 dominates W1, the slope is higher. V2 dominates W2, it's higher. But when I add them up, then the red now dominates the blue. That's the reversal or the paradox. It happens all the time, okay? And the reason it's important in single-cell RNA-seq is because separately from the issue I talked about before, which is correlation between genes, this particular actual same thing, this exact paradox, if you will, reversal, can affect your impression of the change in gene expression between conditions based on whether you look at isoforms or not. Because if you think about a gene that has constitutive isoforms, those are different classes, and if you measure overall gene expression, then you're averaging overall your isoforms. So every time you do an average, to get gene expression from isoforms, you run this risk. And here I've shown you the same thing. You have isoform, 1. You have isoform Y, you have isoform X in condition 1. So you have two isoforms in two conditions, condition 1 and condition 2. And I've set it up here so that the fold change between conditions is reversed when you look at gene expression, namely summing the isoforms, than when you look at them separately. So this is to argue that you need that's why we do single cell analysis and also why we do isoform analysis. You had a question, sorry.
Yeah, so what happened in history is that the Southern Democrats actually moved to be Republicans eventually.
That's true.
So my question is, does that suggest something about the time development of the genes? If you have this paradox, maybe suggest this gene would be, see what I'm saying?
Okay, I think the time aspect here is less, these things can change over time, but if you look at what really causes this paradox, and you can see it in this picture here, but you can also see it in the numbers here, is that, yeah, you're asking a good question, like what causes the actual paradox? And what you can see is it's because in the two groups you have different relative number of people. Like the issue here is that you had only 10 Republicans in the House. And so when you do the averaging, you're like weighting things differently, right? Like and so it's these disparate numbers of people in the different classes that messes you up. So I would say that's sort of the fundamental thing that drives the paradox here. Now, as far as, yeah, it's true that like people switched allegiances to parties over time, and it's also true that in your genes, the relationships between them might change over time, and it's just as important to look at groups, right, to identify those changes. Yeah. Okay. So I'm going to summarize this by an analogy that I didn't come up with, but I came up with part of it. So people like to make a picture like this where they describe single-cell RNA-seq like fruit salad, where you have the individual pieces of fruit, as opposed to a blended smoothie, which is kind of like what you see when you do bulk RNA-seq. But I like put up this picture because I want you to think about that there's two kinds of averaging actually that can happen. One is averaging isoforms into genes, and one is averaging individual cells into bulk. And they produce two different kinds of smoothie. The standard thing on the right-hand side, gene-level bulk RNA-seq, is what people have mostly been doing until about 2015. That's doing both of these kinds of averages, but doing gene-level single-cell RNA-seq still involves this, a smoothie, because you've averaged over the isoforms. So ideally, you'd like cell salad that's resolved at the level of individual transcripts, by which I mean individual isoforms of genes. Okay? Because all the averaging can be unhealthy. Right? I mean, when you blend your fruit, right, you get rid of all that fiber. So, all right. So to that end, and I'm just going to end with this, just then I want to go on to actually discussing single cell. With my now postdoc, but former grad student, Sina Buisagi, on the same data, we published a paper just looking at isoforms, and the link is here. And we show, and again, I won't have time to get into it now, that there's a lot of resolution in the isoforms in these data, not just at the gene level, and you can learn. We actually found cell types that you would miss entirely, or things we think are cell types, but you would miss entirely when just clustering at the gene level. Basically, these sort of statistical averaging problems are present in real data, and we have an atlas at the isoform level, and we'll just leave it at that. So, in the interest of time and to move on, I will just say that if you're interested to read more about these kind of statistical averaging problems, they go under various names: Simpson's reversal, Simpson's paradox. In ecology, it's called the ecological fallacy. And you can go and read all these papers, and there's some nuance here between reversals and paradoxes and so on. But the basic bottom line is averaging is dangerous. Okay? Any questions about that before I go on? All right. So, okay, so we've talked about like motivating partly why you do single cell RNA-seq is to avoid averaging. So what does it entail? Well, you have to, dissociate cells unless you do something like blood. But certainly brain is very challenging because especially human brain cells, very difficult to dissociate. It works much better to do single nuclear RNA-seq, and we can talk about that a little bit. So people want to identify the different types of cells. And, you asked about time, and it's very interesting to look at the trajectories of cells across time. And people want to develop, expression biomarkers for disease states. So that's what people want to do. So what are some of the challenges? They're manifold. And again, today we're just going to talk about two simple ones. And I'm going to bring up one for you in a second, just to give you a preview. But, you have to, the data sets are really large. So it's hard to do the real thing on your laptop. And that's one challenge. There's a lot of statistical challenges, and I'll give examples in a second. There's modeling issues, and sometimes these interact. But there's a lot of technology and a lot of data, so it's actually kind of a great domain in which to get, thinking about any kind of machine learning for neuroscience because these data present challenges for all, that involve a lot of the methods that have been developed. Okay, so at some very, very high level, single-cell RNA-seq, right, is you're getting a measurement of gene expression in a single cell, and so you get a big matrix. But I like to say that single-cell RNA-seq is neither really single cell nor RNA, because the reality is that you don't actually go and look under a microscope and count RNA molecules. In this technology, you're using all sorts of tricks to convert RNA into a cDNA library, which you're then sampling. So you're actually like twice removed from the actual RNA. And it's not really single or cell, because actually you don't get complete information right now for single cells. You get a sampling of their contents by virtue of capture that's not perfectly efficient. And finally, most of the analysis is done by aggregating cells into cell types in a form of averaging as it is. So, but to first order, right, that's kind of like what we're doing. And I'm not going to get pedantic today, but I think these terms are actually important to discuss. Like the word expression, it carries a lot, it does a lot of lifting. It can sometimes refer to the total protein output in the cell, but that's not what's measured here. You can talk about gene expression, but there are multiple isoforms to genes, so it means you aggregated those isoforms or not. It's a little bit like, sorry, I don't know how to turn this off. So the term expression is complex, and the term gene is complex, because it's not entirely clear, even if you were going to aggregate isoforms, like which isoforms actually constitute one gene. Sometimes genes overlap. You're going to have genes containing other genes. And when I said this word matrix, let me just go back here. When I was talking about that table, but this is a math word, and it's actually used to represent a linear map, and that's very important, and Tara is going to talk about that in her methods discussion today, like what this word matrix refers to and why we use it and what value that has. Okay, so, all right, so if we're going to actually talk in detail today about data, it's important to talk about the technologies that produce them. And I can't do it all in the remaining, I guess, whatever it is, 30 minutes or 40 minutes. But I will tell you a lot of the, just for, I think it's some, oh, my goodness, so sorry about this. I really don't know how to turn this off. I apologize. It'll keep going for a while. So there's just so many issues to think about when you do one of these experiments that all play a role in how you analyze the data. And some of these, some things like, so in an ideal experiment, you would assay every cell, and you would have 100% capture rate. You would get every RNA in the cell measured. And you can see my wish list here. Everything would be easy and cheap, but it's not like that. And you often have levers you can pull or knobs you can tune where you can get better capture rate, but it comes at costs, and all these trade-offs are things you have to think about when designing one of the experiments, and you have to also think about them when analyzing them. And so I'm going to show you some of those. I just want to say, by the way, that there's a whole universe of methods now, including many spatial methods, and we wrote a review in our lab with one of my students, Lambda, recently. I'm not even going to talk about any of these, and they're actually really, really important for neuroscience. But that's just how it is. So we're doing one day here. Also, there's like huge amounts of data coming. This is from a former postdoc in our lab. He put together a database, which he still updates. He takes every paper that comes out and puts it in this database. And he's tracking how many studies there are over time. And it's just like, this is already like a bit outdated. This plot, which I really like, is one that's made with this notebook. You're going to use Google Colab this afternoon, and I ran it this morning. So this is up to date. There's another database that is collating all the tools that have been developed to study single-cell RNA-seq. There are now, I think, around 1,200 tools. Each of these is like a paper saying, like, here's my new program for you to use to analyze your data. So you can see that there's a relationship, like, about 3/4 of a tool per paper. Okay, so Yeah, I see people shaking their heads, right? It's kind of overwhelming, right? So like, if you're, so working in this field is really hard, actually, okay? Because which tool here do you use, which method? Reading all of this is obviously impossible. By the way, and we'll talk about this more later, but this Colab tool, which you're going to use or have already in this workshop, is super cool because, like I was able to like remake this plot using this notebook and you're getting these slides, so you can click on it and play with it yourself. And because in the notebook you can see the links to the databases that are being like scraped to do this, so it's very transparent. So one thing I want to just tell you up front is that lots of tools don't translate to lots of understanding. And I want to do this by way of showing you just like one of the most simple and first steps in any analysis, quote unquote simple, is to normalize the data. And I'm going to show you right now how complicated it is to decide how to do that. And so to show you that, I'm just going to first show you a little bit of data. And I just have 2 slides on this, but I'm going to show you like a conundrum or a problem that is actually very, very challenging that a lot of people sweep and ignore under the rug, but like just shows how complex it is to actually think about any data set or your own. So in this plot here, this is from a recent preprint, not from our lab. And it's just showing like different data sets. So, let's ignore the top panel here, but down here, these are various cell lines. And so it's like super simple. It's just a cell line that's been assayed by single cell RNA-seq. And every dot here is a gene, all right? And what you're seeing is, on the x-axis is the average gene expression for that gene in that cell line. I mean, there's not even cell types here, right? These are like cell lines, right? So it's like super simple. So, but there's some variation here, right? Why is there variation? I mean, it's life. Like, you're doing an experiment, right? Like, it's not like so there's variation. And you can see that some genes are sort of highly expressed, you might say. They have a high average expression. Some are low. And on the y-axis is the variance, okay, that you're measuring for that gene. And what you can see if you, so this is, again, philosophy discussion that Tara's put together, talks about exploratory data analysis. That's going to be our topic today. This is a typical kind of plot you should make with your data always to just see what's going on. Because it shows you something about the relationship between how much variation you have across your cells for a gene based on the expression level. And what you are seeing here is that, well, there's obviously a relationship, but the relationship is what's called superlinear. It goes above the line y equals x. And to understand this plot requires just a very little bit of math. And the first is to understand that, like why would you have a relationship between variability and variance, okay? And maybe I'll just leave this slide up, and I wish I had a whiteboard here or a blackboard. I don't. But there's sort of 3 math steps you need to go through, and I'm just going to say them out loud, but you can talk about them later. The first is that if you perform what's called multinomial sampling, like you have a bag with different colored marbles, right? Those marbles that are different colors are your different genes, and you just pick up things at random. You're going to have some variation in how much red you picked up every time or how much green. Your bag might have a lot of green and a little red, and you might be able to figure that out because you tend to pull out more green. But the exact amount of green you pull out is going to be variable each time you do an experiment and reach 20 times into the bag. And that is called, the math that models that is called the multinomial distribution. And it's well approximated by another distribution called the Poisson Distribution, which you may have heard of. And if I had a pen here, I would write it out. I have crayon, and I can write it for myself here, but then you guys can't. Okay, you guys can't see anything, right? So it's useless. Yeah, it's okay. We'll do it later. I mean, Tara's got paper for all of you, and we can do it this afternoon. Maybe I'll walk around and we can do it more one-on-one. Yes, you have it in your slides, right, in the methods as well. Yeah, exactly. So, in any case, under this sampling model, just randomly sampling out of a bag, just take my word for it, you expect to get data that looks like this, where the variance scales exactly with the mean. In fact, it's equal to the mean. So you would see points straight on this pink line, okay? So because the experiment is random and you're sequencing cDNA molecules out of your library by random, you expect to be on this pink line. But what you actually see is in these cell lines, you go over the pink line, okay? It's a little bit more. And to a statistician, that says, well, my experiment was more noisy. Maybe because things aren't perfect, okay? There's a lot of other stuff going on in my experiment other than perfectly sampling molecules out of my cDNA library. Maybe in some cells, I'm a little bit better at sampling than others, and that variation would just increase the variance here, okay? All right. So here's a question for you. And I'm not going to answer it for you right now, but you're going to look at it a little bit this afternoon, but it's a good question. So there's like this variability in your data that's a little bit more than you expect usually. Is it extra technical noise? Or maybe is it because of the fact that actually in your cells, genes are actually expressed variably? I mean, you know, gene expression isn't this like deterministic machine where it's like, you know, on, make gene, off. There's variation in stochasticity in the cell, right? Transcription factors have to bind. Sometimes they bind, sometimes they don't. So which of these two things are you seeing in your data? And that's kind of actually important to know, because if you believe it's all technical noise, then the first thing you do is a step called normalizing your data, and you remove it, okay? But if you're a biologist, you might actually want to know whether there's variation, like some cell types are just more variable than others because the transcriptional machinery is more variable. So this is a basic question, and this distribution, which again Tara is going to talk about, called the negative binomial, which gives a quadratic curve above that pink line. It's widely used. Computation, like right now in the field, most people assume the data is like that purely due to technical noise, and the first step they do is clean the data or normalize it and remove all that variation. But there's a whole bunch of people at Caltech, people like Rob Phillips, and there's others, who actually have worked out what mechanistic models for the cell imply about the variation, and they get the same distribution. So by their models for what goes on in the cell, all of this variation is purely biological, and you shouldn't touch your data at all. You shouldn't clean it. So, you know, so here's a question for you. Like, you got your new data set, it's nice and shiny. Do you clean it or do you not clean it? And it's actually, if you do not clean it, your reviewers will say, why didn't you clean your data? Everybody cleans their data. Okay? If you do clean your data, some of your reviewers will be like, well, you just threw all your baby out with the bathwater, all your biological signal. What are you doing? So, okay, it's a problem. Okay? Yes? Well, so before, that's a great question, and I'll tell you that it's very, like, before one can answer that question, one has to have a method for looking at your data and figuring out what's technical and what's biological. And out of the 1,300 methods, there aren't really any methods that actually will do that for you right now, unfortunately. So, okay? All right. So I'm the bearer of bad news. But the point here is not to teach you how to do analysis, but just things for you to think about. All right, so if you're going to like actually know whether the noise is, you know, whether the variation is technical or biological, you actually have to understand also how your experiment works, which is, I mean, again, I don't have time to go through all the details, but I'll do some. So here I've listed like a variety of protocols that are popular. Some of them are not so popular anymore. I mean, this changes like every couple of months. Right now, lots of people use a technology called 10x. But actually, I'm involved in several projects that use split-seq, which is all the way on the right. 10x Genomics is a company that does single-cell RNA-seq. And each one of these technologies, there's different choices that are made and different methods that change completely the profile of the data and the noise structure and how you would analyze it. So, you know, each column here requires a complete, like, PhD to figure out, like, what you should be doing with the data. So, but at a high level, there are methods that separate cells into individual wells. And then, there are methods, and this, I have too much stuff, but this is the protocol for one such method, SmartSeq2. These methods are characterized by the ability to have very low cell loss. Because you're putting cells into individual wells, you can capture most of your cells. But they actually have throughput issues because you have to have either, standard 384 well plates are not going to get you that many cells unless you have a robot to do, dozens of plates. Some people now use what are called micro-well plates where they can put, thousands or millions of little wells on a single plate. But anyway, these methods tend to have high capture, lower throughput, and there's a lot of different trade-offs with them. Cost, by the way, is very complicated to measure. And again, why is it complicated to measure cost? Because there's sequencing cost. And for example, in this method, SmartSeq2, you actually sequence reads from across a whole transcript at a time, and not just from the 3' end, which is what's done with other technologies. So you pay a lot more sequencing cost per transcript you're measuring. That's good because you might get isoform resolution and not have to deal with the averaging across genes that I showed you can be a problem. But on the other hand, you have to pay a lot more cost. So I'm giving you this sort of basically just to have you realize that if you're actually going to use these techniques, you need to think about that. And we're not here to decide technology. We're in a data analysis AI workshop, and the AI methods you use are, they're going to depend on exactly what this data looked like or where it came from. It's very important. Even just sequencing costs are really complicated and play into the decisions upstream of getting your data, because it turns out that sequencing large amounts in bulk is cheaper, but then you have to have more data in one go to sequence. So, so there's lots of trade-offs like that. A separate kind of technology, or different one, is Droplet-Based. And this comes from the development of several lines of work. And I won't go through the whole history, but two of the main open source methods that were published were done actually in the same issue of the same journal in 2015. They were called Dropseq and InDrops. And they come from methods from David Waits' lab at Harvard and actually Rustem Ismagilov, who's now also at Caltech. And these methods involve creating basically aqueous droplets in oil that look like this, in which you're going to capture individual cells and beads inside droplets. And you should think about each of these each of these droplets as becoming your mini chemistry laboratory, if you will. So you're sort of parallelizing the assay by creating lots of mini labs. The InDrops approach is, I took this from this paper, involves using microfluidics device to pinch off these droplets. Inside each droplet, you're going to try to capture one cell with one bead. And the bead is a hydrogel bead that has on it DNA oligos that have poly-T ends to capture polyadenylated transcripts, okay, that have poly-A's on their end. They also have barcodes on them. You can see that here. So a single bead will have all the same cell barcode so that when you do sequencing, you'll be able to recover which cell you were from. But they'll also have, each oligo here will have a different UMI, it's called, or unique molecular identifier, so that you can distinguish molecules from each other. So that when you do PCR later on, you know that it was one molecule and not 50. So it's complicated, but at a high level, it's simple. You have these aqueous droplets. They look just like this. And inside each of them, you try to capture one cell and one bead. And I have to tell you this if you're going to learn how to analyze the data and use machine learning, because I'm going to show you in a second that sometimes you get 2 cells or more. Sometimes you get 2 beads. And you have to be aware of that. Otherwise, your analysis is going to suffer for not accounting for those kind of issues. I'm, again, not going to go through the protocol, but I want to tell you kind of at a high level, what can go wrong and what needs to be accounted for in the analysis. So your goal is to get one cell and one bead inside one drop. Oftentimes you'll have one bead and no cell, and you don't care about those because there's going to be no data there. So those are irrelevant. But all those scenarios are bad, and you can enumerate them. A doublet is when you have two cells with one bead. Why is that bad? Well, because that bead has this one cell barcode on it, and when you do your sequencing, you're going to think that all the RNA that was actually from two cells was from one cell. So you've done a kind of averaging that you didn't intend to do. You can actually have a cell with no bead, and that's bad because you just lost data. All right? You can have what I call a split when you have one cell and two beads, and now it looks to you like you had two cells, so you kind of have a duplicate of your data. And you can have a collision sometimes where you have, because of the variability in the sequence on these cell barcodes, you might just by accident have the same barcode on two different beads. All of these things can happen, and they do happen. And part of the data analysis here is to clean out the data, is to like figure out when all these things are happening and understand why they're happening and what to do about it. And I'm just gonna like, I don't want to go too much over time, so I'll try to cut things short a little bit. But all of this is just like math. I'll show you. So, you need to calculate like the probabilities of these things happening, and then you need methods to find them. And sometimes the solutions lie in the machine learning, and sometimes the solutions lie in the experiment design. For example, if you load a lot of cells onto your experiment, you'll tend to get more doublets. So you can reduce the number of cells. That's something you control as a user. So the answer isn't always to use machine learning to analyze your data. Sometimes it's to produce better data. All right. So what kind of calculations do you need to do? Well, you do like math like this, and I'm not going to go through the details with you, but it's kind of fun. If you have n cells and you have m barcodes, and you imagine that you have a cell and you're picking like a barcode at random to pair with it. If you don't have a lot of barcode diversity, you're going to have these barcode collisions. And you can calculate like the expected number of collisions, and you get formulas like this. And I'm going to skip over this, but it's basic math counting to get these formulas. Yeah, and I think we're just too short on time to do all this, especially starting late. But I think you just take my word for it that these formulas allow you to assess how much barcode diversity you need when you build your experiment. They also allow you to assess whether you need to worry about this issue in cleaning out your data as step one of your whole analysis, because you can actually plug in for what this number is, or you can estimate it, and then you can figure out if this is an issue. All right, so I talked about barcode collisions. There's a lot of tuning concepts. I mentioned before the capture rate. I've mentioned the split rate. I've mentioned the doublet rate. And what I kind of want to say is that each of these things involves trade-offs. So maybe I'll go back to this picture here. You can reduce, for example, the doublets by loading fewer cells. Then you have like a lot more cell loss, okay? So there's lots of trade-offs, and maybe I'll try to go through some of them here. I think, okay, I'm going to skip this, but maybe I'll show you this plot just from some food for thought. So you're loading cells into droplets, and there's a rate at which you load them. So, in these microfluidic setups, including the 10x genomics, there's sort of like some randomness in whether a droplet gets a cell or not. And this particular distribution, and now you can see its formula, tells you the probability that you'll have 0 cells, 1 cell, or 2 cells in each droplet. You can also tune the bead rate so that you have either 0 beads, 1 bead, or 2 bead. And what you want to do is you want to have this be pretty high, because if this is too low, you lose cells, right? You don't capture cells. On the other hand, if you make this too high, you get more than one bead inside a drop. It's the same with this, right? You want this to be high enough that you get your cells captured with beads, but you don't want this to be too high, because then you get doublets. So, okay, so these are kind of what you have to tune. And you can make plots like this, like how your capture rate varies with your split rate based on like these choices you make. You can't win, okay? Like when you have this randomness, you up this, you destroy that. But you got to find a trade-off. And in analysis, like step one basically is to look at your data and actually estimate these things so that you can get a handle on, on like what was going on in the experiment, so that you can understand if something went horribly wrong. Now, I keep saying this, and it's really true that like, learning machine learning for data analysis cannot be decoupled from like doing the experiment, because oftentimes you can, improve your analysis prospects by changing your experiment. And this is very true in these microfluidic setups, because it turns out that the bead material really matters. So in Dropseq or there's polystyrene beads, InDrops uses hydrogel beads. And you might go, what the hell does the bead composition have anything to do with your data analysis? Well, the thing is that hydrogel beads, they can compress and they can be packed tightly together, so that when they're injected into these aqueous droplets, you can actually control to much better get one at a time. They have a real advantage. And you don't have as much randomness and variation in the number of beads per droplet. So technologies that use hydrogel beads are better in this sense. And that's one reason 10x genomics has become popular, is because it uses hydrogel beads. And you can see this here. The beads are tightly, densely packed up there. As they're getting shot basically into this flow, you have single individual beads getting captured per droplet. And 10x genomics has done a good job of optimizing the chemistry of their beads and the flows to make sure you don't get too many splits, right? Okay, now I've already, sorry, let me try to get this. I really don't know how to get rid of it. I've turned off my volume everywhere. I'm sorry, guys. Okay, so now, this I've already mentioned. The doublets occur when you have two cells getting captured in a single droplet. And you can control that by decreasing the number of cells you load onto your experiment. On the other hand, and that's really, and so this formula here, can be derived, and it's a really important formula because it tells you how many of such droplets you expect. And you can measure these in what's called a Barnyard Plot. So this is going to be also interesting for the philosophy discussion. This is a plot with X and Y axes where people have loaded two different species into one experiment. This is useful because you can now identify, oh my God, this is horrible. I really don't know what to do. I'm so sorry. I will scold my kids tonight. All right. It's my kids. I apologize. I've quit the app out of my phone, out of my computer, but somehow it's still like, you want to help me? All right.
Yes. You see this?
Yeah, it's off.
What? Oh, yeah.
Yeah. It's off, and I've turned the sound off.
Have you tried turning off the phone?
I have not. Yeah, let me try to turn off the phone. But I think it's actually coming from the computer, not the phone.
Oh, in that case.
Yeah, so. Now on the computer, I quit the app entirely. I mean, I can turn off the whole computer. I can't mute the computer because I'm on Zoom. You think this is easy, but I'm like, I'm a computational biologist. It's like, no, and I can't even do it. No, they want to make sure I can't ignore my kids. All right, I'm sorry, everyone, for the diversion. But all right, so where was I? So yeah, so you can control this kind of thing. in these experiments, which are really, really important controls to do, prior to analysis of your data, you actually run an instrument, but you can actually do this in other ways, even within an experiment, where you can actually tell when you have a doublet because you have two different types of RNA. You can do this by loading two species into your experiment. You can also do this by tagging your samples with some other barcodes and then seeing them in the same cell. And you should get the data on the axes here because, in this case, you're either looking at, this species versus that species. The purple are when you're detecting both in one droplet. Okay, so this is useful. A very basic like cleanup procedure to do is to look for doublets like this. But if you don't have different species or other kind of labeling, then you have to guess like what the doublets are, and there are a lot of tools that do that. I will point out one thing that, and this is where the math comes in again, and why we need more than just an hour to really do all this properly. But even in an experiment like this, the purple are doublets, but there are doublets that are not purple, because you can have two red ones that are a doublet, and you can have two blue ones that are a doublet. And there's a nice paper that works out the math of how to estimate how many of those doublets you're also seeing in your data, just from seeing the purple ones. And that's really useful, because this adjustment factor can tell you whether you need to really worry. So you might see like 2% doublets or 5%, but actually you're higher, like maybe you're 8 or 10. And so this adjustment is a very nice adjustment, and it's in this paper by Jesse Bloom from the Hutch Institute. Became very famous during COVID now. All right. Now, technical doublets are when two different cells get captured in one droplet, but there are also biological droplets, which are different, which are actually two cells are just sort of clumped together and don't dissociate. And now your problem isn't that you had stochasticity in your technical aspect of your experiment. You literally have just two cells that are still a doublet, but for different reasons. And so there's different ways to deal with that. One of them, and that's what for neuroscientists has been a great approach, and because since nuclear single-cell RNA-seq is easier to implement for human brain cells anyway, it has the advantage that you don't tend to get these biological droplets. But anyway, that's another issue I wanted to raise. And so in summary, you have all these different issues. You have these knobs you can tune, the rate at which you pop in cells, the rate at which you pop in beads, and the different choices affect all these different artifacts you might see. And as I said in the beginning, in the absence of figuring out all these artifacts, you don't even know how to perform step 0 with your data, which is how to normalize it. Now, there's a whole other layer of complexity for single cell that you're going to see this afternoon. And that is that, there's unique molecular identifiers, which are DNA sequences that also get sequenced, but that are supposed to allow you to compensate for PCR steps when you're making your cDNA library. So that if you had one molecule in an actual cell, even though you might see it 20 times in your library, you know that it's just amplification of that one molecule. But again, the UMIs have some kind of diversity. Usually they're pretty short, and so you might have collisions where you have the same UMI on different molecules. You have to worry about that. And there's a lot of learning to do into how to actually deal with all of that. I've said, that some these assays like 10x's assay or indrops only look at the 3' end of genes. But you shouldn't take that to mean that you only get data right from the very, very end of the gene. There's actually a distribution where you might see several thousand base pairs into the gene just by chance because of the way the fragments sizes end up scaling. So there's actually a distribution on how far from the end you see data. That's really, really important because that means you get better access to some genes than others. So, one of the analyses we talked about last week was PCA. And this kind of stuff really matters because when you do PCA, you're making a lot of assumptions about your data, namely that each gene has equal variance and that each gene was assayed to the same extent. If you have a gene which is very long, then you don't have as much access to its material than a short one. You're going to have biases there, and that's in turn going to affect the variability in the gene, and that's in turn going to affect the PCA. So, all right. Okay, in addition to all of those issues, you have errors in sequencing. The actual sequencing may not be, faithful to the actual barcodes that you were trying to look for. So you may think you saw one barcode, but actually seen another because you had a sequencing error. And there's ways to deal with sequencing error correction. So this stuff is not really AI, but it's necessary before you do your AI. And if you don't think about it, then what comes downstream is kind of junk. So I put a last link there to, this is now a couple of years old, but it's helpful. It's a paper that summarizes all of the main aspects of each of the different technologies and their strengths and weaknesses, like SmartSeq and 10x are two main ones, and sort of summarize some of what I've done in this whirlwind tour this morning. Okay, so I think I will, it's now 10:55, so in the interest that we don't get way delayed today, I think I'll stop here. It's okay.